So I've got a lot of slides, but I'm gonna skip over a few because I want to get the main messages over, and I know we're running a bit behind here. So I'm just going to talk a little bit about the microbiome in IBD, talk about fecal transplantation, which has been on my mind really day and night for the last 3 years, and then other types of IBD which are also interesting in context of the microbiome. So just to get this one over with, really, so there's a microbiota and microbiome. Okay, the microbiota is all the bugs in a particular niche. In this case, we're talking about the gut. The microbiome is all the genetic material, which is, but essentially you can use the term interchangeably, and it is used interchangeably in the literature. So this is the narrative, really. It's about the association between the reduction in chronic inflammatory conditions in the 20th century associated with the use of broad-spectrum antibiotics, and the association with the increase in chronic inflammatory conditions. And the narrative which plays really is which is gaining a lot of traction, is that this is a result of a disruption of the microbiota in the gut in the Western populations in particular, where the chronic inflammatory conditions are a big deal, really. Of course, we now know that the bugs in the gut are equivalent in number to human cells in the body, and these are not just passengers, but intimately involved in nutrient digestion, so B vitamin metabolism, short-chain fatty acid production, which is really becoming very key. It seems that the short-chain fatty acid production by bugs which can digest the resistant starch, which we have difficulty digesting, is really key to gut health, epithelial homeostasis, and particularly to the development of the mucosal and systemic immune systems, in educating the immune system that actually the microbiome is a hazard or should be tolerated. That balance is what it's all about, really, because the gut is the biggest interface between the environment and the body's immune system. So these are not passengers. So just a quick word about taxonomy. So very boring subject. I kind of fell asleep in medical school. I'm to learn it again, unfortunately. This is the taxonomy of a human being. So we all belong to the kingdom of animals. We have backbones, so we're chordates. We're mammals. We belong to the order of primates, family of hominids, genus of Homo, and the only species around is Homo sapiens. Most of the literature in terms of the ecology of gut microbiota uses 16S ribosomal sequencing, and that gets down to about family, at best genus level, okay? So you've got to bear that in mind, because a lot of the literature is based on a very high taxonomy, and some of it is reported at sort of phylum level. So I hope there's more to me than just a backbone. I mean, it's kind of, we have to just bear that in mind. And just that word on methodology again, so most of the literature still is looking at 16S RNA gene sequencing, which is a specific variation of the genetic code in ribosomes, and that allows you to get to sort of about the family genus level. The best way of doing this is by doing metagenomics, where you break down all the DNA in a sample and analyze that to try and get the whole gene sequencing. We've forgotten about culturing, because this field has been driven by non-culture methods, because it's very difficult to culture bacteria from stool, because stool live in an anaerobic environment. Once you pass the stool sample, you can't culture. So all this literature has been driven by non-cultured technology, but we're beginning to realize that if we combine careful culture with metagenomics, then you get the best data out of any samples. So the main problem with 16S sequences, you can't, for example, differentiate between multiple resistant or sensitive Staphylococcus aureus, for example. So we're now focusing more on metagenomics, and this is some lovely data from my colleague who I'm working with in the fecal transplant studies. He was called over to look at the latest Ebola outbreak, and using metagenomics was able, for example, to track the strain of all these cases to one survivor in whom those strains were present. So very powerful technology, and we should be sort of focusing on this now. So what's normality? The human phyla, the human colon consists of 4 phyla, so at a high level. There are the firmicutes, which are involved in harvesting energy. 35%, about 30% of the gut is of firmicutes. The bacteroidetes, gram-negatives, about 60%, and they're involved in the degradation of complex sugars and proteins into short-chain fatty acids. So these are the good guys. The bad guys are the proteobacteria. So these are gram-negative bacteria, which are mainly the pathogens which take over and cause disease. For example, E. coli, the Salmonella, the Shigella species, they're all in the proteobacteria. And then there's actinobacteria as well. So generally, the sort of message that's coming across is that the more of this stuff you have, the better you are. The less of this, the more of this stuff, the more damaged it is. And the more diverse your microbiome, the better. The more sort of families and species there are, the better. Of course, the microbiome changes from birth to death. So the uterus is not sterile, but infants derive their microbiome at birth from the birth canal, and the method of birth is really important in this case in terms of defining what that is. And then from the birth to the cradle, there are numerous genetic factors and environmental factors involved. So, for example, if you, depending on your diet and the drugs you take, so the environment is much more important, really, in determining the microbiome than host genetics. Drugs have a major effect, but generally speaking, from adolescence into late adult life, the microbiome is very stable. So even if you give healthy individuals a course of broad-spectrum antibiotics, within 12 weeks their microbiome reverts to its normal state in health. As you get older, your microbiome becomes less diverse and the balance changes as well. And diet is really the key, and diet is very missing in a lot of this literature, and I wanted to kind of just emphasize that all the way through this, really. Here's one diet study, which is a healthy volunteer study, where patients, these healthy volunteers were changed from a animal-based, typical high-fat, high-protein diet to a vegetarian diet high in resistant starches. And it was found that when changed from the good to the bad diet, there was an increase in bile acids, a change in carbohydrate to amino acid metabolism, and increased sulfide-producing bacteria on the high-fat, high-protein diet when it was changed from the good to the high diet. And that was within 1 day. Within a day, within 24 hours, the metabolites in the stool reflected the change from good to bad, and also the genes were also reflected in that change within 24 hours. A very rapid change. If you compare healthy individuals living in Africa compared to Europe, then the Africans tend to have more in the way of bacteroidetes, so again the short-chain fatty acid producing carbohydrate and fat and protein metabolizing bugs, whereas in the European populations, more firmicutes, more in the way of energy harvesting from the diet. And this is a really interesting study focusing on colon cancer a few years ago from America, where they looked at the diet of American Africans compared to South African native Africans. And they found that, we all know this, that the American African diet was much higher in protein and fat. There was more proteobacteria, more Escherichiae, so E. coli type species, Acinetobacter, and increased bile salt degradation as well. Bile salts, secondary bile salts, are highly carcinogenic and not good. Whereas in the African diet, at baseline, there was more high fiber starch. There were more bugs in relation to starch degradation, carbohydrate fermentation, and butyrate. And when you switched, there was a remarkable change from one to the other. And this was reflected in reduced inflammation in the colon in these populations when they switched from the bad Western to the African diet. And also, actually, very interestingly, in neoplastic markers as well, markers of cancer. So that was really interesting. So in IBD, in IBD, right from the first metagenomic analysis of the genes of, in the stools of patients with IBD, there was a clear clearly distinguished between healthy ulcerative colitis and Crohn's disease individuals in terms of the microbiome. And the signal is not surprising because in the end, IBD is all about a genetically driven predisposition for the environmental triggers to trigger an immune response. And the microbiome has got to be key to that. The epidemiology of IBD reflects the fact that the environment is really important. So 40 years ago, it was a Western disease of the 20th century. Now, my colleagues in the Middle East and India are seeing just as much IBD as you see in New York and so forth. So that cannot be a change in the genetics or the immune system. It's got to be an environmental trigger. So the environment, and particularly the microbiome, is a really fascinating area in which to intervene in the context of inflammatory bowel disease. So again, just in IBD, you see generally a reduction in the good guys, so the firmicutes and the bacteroidetes, those involved in short-chain fatty acid production, and an increase in proteobacteria, and a reduction generally in the diversity of the microbiome in the colon. And there are some specific differences in Crohn's disease and ulcerative colitis, again in relation to reduced firmicutes, increased E. coli, the proteobacteria, and in ulcerative colitis here again, very important, these species which are important in short-chain fatty acid production are reduced. And these short-chain fatty acids are really important in the gut in terms of a very complicated slide. I just want to focus you on this bit here because this is the key to IBD, where the T cells, which drive both the innate immune protection against infection and also tolerance, are driven either towards the T17-TH phenotype, which is causing inflammation, or the T-regulatory T-suppressor phenotype. In IBD, we're going this way, so you get more of the sort of inflammatory phenotype compared to the suppressor phenotype. So Tregs go down, and these T helper cells go up. And all the treatments for the pharmaceutical treatments for the treatment of IBD are all focused on these pathways. All these monoclonal antibodies, all the immune suppressants, are all about reducing the inflammation as a consequence of the overexpression of these T cells. So, manipulating the microbiome, the actual trigger, seems like a very interesting and valuable thing to do here, possibly. So, there are three ways of doing: probiotics, fecal transplants, diet. I'll put that in brackets because nobody's done it properly, right? So I don't normally quote Kipling, but this is for Professor Barton. It's just to show a bit of literature. Yeah? So probiotics are useless, aren't they? Is that door shut? In IBD, they've been very disappointing. They are kind of helpful in maintaining disease. In patients who, if you treat them with 5 ASAs for IBD, and also if you have pouchitis, then there's a small benefit of staying on probiotics to prevent recurrent pouchitis. But basically, it's the law of the jungle. The strength of the pack is the wolf, and the strength of the wolf is the pack. If you transplant the whole microbial community with all the then you're more likely to get an effect than if you just choose individual strains, which is what probiotics are. It'll get better, but it's not good at the moment. So the concept of fecal transplantation, which is what I want to go on to now, is that patients with dysbiosis or an imbalance in the bowel, in the microbiome, have lost their healthy microbiota, and this can be theoretically corrected by the transplantation of stool from healthy screen donors. And again, just to reiterate, there's this recognized dysbiosis in inflammatory bowel disease. Fecal transplantation has been around since the 4th century, so in traditional Chinese medicine, known as yellow soup, and also in veterinary medicine. And in the 20th century, it was first used by an American physician to treat patients with pseudomembranous colitis, which wasn't recognized as being related to Clostridium difficile at the time, but clearly was. And in 1958, these soldiers with pseudomembranous colitis had fecal transplants and did really well, dramatic response. It was then forgotten until a gastroenterologist in Australia decided to treat himself in 1989 and he was off medication for 12 years. So it kind of caught on then. If a doctor's going to do it to himself, it's got to be good stuff. Australians do this all the time. They give themselves Helicobacter pylori, fecal transplants. In C. diff, you must work. C. diff is a devastating condition in which patients, particularly elderly patients, are given broad-spectrum antibiotics, and they get this horrible debilitating diarrhea. Antibiotics work about 70% of the time. 30% of patients get recurrent C. diff. And if you look at this meta-analysis we published a couple of years ago, these are the case series here of treating patients with C. diff colitis with fecal transplants, and these are the randomized controlled trials. This is 90% cure here. So you can see it's highly effective treatment, and it's really quite a very rewarding thing to treat patients with fecal transplants who've had recurrent C. diff. So on the basis of this, we set up a lab in Birmingham and then had to get an MHRA license. This is the only licensed lab to produce licensed stool as a medicine in the country, and we're treating now thanks to these guys from Blood Bikes. We've been sending this around all around the country. So if anybody needs fecal transplants for C. diff, you can get it free from Birmingham. That's the advert done right now. In ulcerative colitis, there have been 4 randomized controls trials so far of treating in ulcerative colitis with fecal transplants. And this is a busy slide, but I just want to point out the methodology was very different. So the first trial just used enemas for 7 weeks. This trial here used nasogastric treatment, nasojejunal treatment, but used patient's own stool as a placebo. This was 2 Australian trials where they used mashed up stool from between 3 to 8 different individuals and gave it in a fairly intensive way through the colon. And so pretty diverse methodologies. The follow-up really is quite short. So it's supposed to be 12 months, but it didn't really happen in this trial. And so you can see the maximum follow-up is 3 months. But the remission rate for fecal transplantation is about, in these trials, the second one, the one where they used nasojejunal treatment from Holland, didn't work. The other trials showed met their primary endpoint, and the remission was about 30% compared to about 9% in the placebo groups. And that doesn't sound great, but that's the same as the TNF-alpha remission in the seminal trials in inflammatory bowel disease. 30% remission is what you see in all phase 3 trials compared to placebo with these immune suppressants. The donor matters, obviously. So from the first trial, this lady, donor B, was responsible for the trial reaching its endpoint. When she started taking antibiotics and came out, the results weren't so good. So this is the lady with the golden poo. Trouble is, we don't know what that is, that golden poo. From the ulcerative colitis trials of fecal transplantation, there's definitely an efficacy signal. There are early signals relating to donor effect. And these are the richer the donor stool in terms of diversity, the more Clostridia there are there, which are firmicutes, then the better. And there are early mechanistic signals in terms of the recipients, in that the recipients who do well have an increase in diversity and increased short-chain fatty acid production. We don't know which is the best route to give the stuff in ulcerative colitis. We don't know what makes a super donor. We know nothing about diet. The word diet isn't mentioned actually in those 4 randomized controlled trials, which is quite amazing, really. We don't know whether engraftment is happening, so whether the result of the microbiome changes in the patients who get the fecal transplants as a result of the actual engraftment of species from the donor, or whether it's just a recrudescence of species which have been suppressed by the inflammation. We don't know about host factors, and we don't know how long the effect lasts either. So we are halfway through this randomized controlled trial. We've done a pilot first to actually look at the route, try and characterize a good donor based on EPIC questionnaire diet data, look at the metabolic consequences and safety. So we've finished the pilot. I can't show you the full results because we've got to wait for the independent oversight committee, but I'm just going to show you our first patient that was treated by the colonic route. So this is what the bowel looked like at the beginning. and we gave 8 weeks of this fecal transplant, and this is how it was at the end. So, I mean, we've seen some great results, but I mean, this is not a randomized controlled trial, it's open label, but there's a nice efficacy signal. Happily, that was mirrored in increase in gut homing Tregs in these patients who responded as well. So indicating that the microbiome might be affecting the immune system in the right direction. We don't know how it works. We're using strain tracking at the moment to track between donors and recipients. It may be that we are replacing bacteria with good bacteria, Maybe we are replacing the bacteriophages, which actually outnumber, so the bacterial viruses outnumber bacteria, and these are killing the pathogens. Maybe defensins produced by these bacteriophages. It may be microbial metabolites. Really, everything is up for grabs at the moment. I'm going to skip over this, but just to say mice aren't men, and even the mice immunologists are going off mice models in this space. This is what's known. The cupboard is bare in terms of the information about diet in this space, in terms of FMT. I just want to reiterate that. There's been some early work now. So this was presented by Ari Levine at ECHO. I think you're on this one. You didn't know. So this was supposed to be a project of diet manipulation combined with fecal transplantation ulcerative colitis. So I'm sure you've heard of Ari Levine. He's a leader in this field. He was going to look at how his particular diet improved colonization in the gut, how it improved remission, and also how diet improved the microbiome. I'm quite reassured to hear that he had great trouble getting this going with regulatory things. So it's taken 3 years to get the regs sorted out. So he didn't present any data from the treatment of the ulcerative colitis patients, but he presented donor data. He put the donors on 2 weeks of increased fruit and veg and resistant starches and reduced animal protein and fat. The details of the diet weren't available. And he was looking for a reduction in the bad guys. So these proteobacteria, the bile salt degrading bacteria, and an increase in the short chain fatty acid producing good guys. And it didn't work in the 9 donors because they were too healthy to start with, he says. The medical students. They had very few bad guys to start with. The diet may have been too simple. I don't know the details, but my usual medical students are sneaking burgers all the time. I'm not sure what's going on. But it's interesting that the work is going on. It needs to carry on. I've seen this paper from Gastroenterology from colleague Dr. Gerasimedes He's also, this is a nutritionist who's into metabolomics as well, and he's designed a diet to replicate modulin as an enteral nutrition, which I'm sure you're all familiar with. And this is an example of the kind of stuff here. So this is chicken sandwiches, chicken pasta bake, chicken with mashed potato and cod and rice. And this is very complicated, but essentially I'll skip over it. He basically put healthy individuals on elemental nutrition or CD, this is his diet. He'd replicated the same thing in mice with colitis, and he treated 5 children with active Crohn's disease with this diet for 4 weeks. He was trying to show that the change in the microbiome response in the mice would be the same as in the healthy individuals, and this is a pilot look at treating Crohn's disease. So essentially, the CD treat diet was well tolerated, better tolerated than the elemental nutrition. There were similar changes in the microbiome with both the adults and the rats, but these, for him, were paradoxical changes, not for me, because the diets were enriched in proteins and fats and low residues. So we were seeing, actually, reduced good guys and more bad guys, increased sulfides and reduced short-chain fatty acids. So this is nice because it kind of confirms that proteins and fats are bad, and the short-chain fatty acids producing resistant starches are good. And you can see here there's a reduction in firmicutes and an increase in E. coli in both the healthy individuals on both interventions. But paradoxically, the 4 kids he treated, 8 of them, 5 kids, 4 got better with Crohn's. So again, that's that thing that Nick was saying, that you've got to be careful about symptoms and inflammation. Just like in celiac disease, it could be these patients still with Crohn's disease, very difficult to assess, may actually still have ongoing inflammation. Their symptoms are better on this low residue thing. So there's a lot to be answered here. Again, I just go back to this, that we know very little about the role of diet in donors for fecal transplants. Checkpoint inhibitors. Checkpoint inhibitor colitis is a big problem in patients being treated for cancers. Checkpoint inhibitors have revolutionized the treatment of cancers such as melanomas, small cell lung cancers, urology cancers, colon cancers. And these work by blocking the inhibition of the sort of T cells which fight infections and tumors. And if you take the block off, then you get more aggressive T cells which fight your tumors. But the other side of that is 40% of these patients get colitis. Okay, and there were 3 science papers last March which suggested if you look at the microbiome at baseline of patients with before they start these checkpoint inhibitors, then it's very predictive of who's going to respond. And actually, if you did fecal transplants from those patients, then the patients who responded well also did well in a mouse model of checkpoint inhibitors for treating mouse models of cancer. And there was a great deal of interest in this. The other problem then is the colitis that goes along with it. These patients get bad colitis, they'll get immune suppressed, steroids, TNF-alpha antagonists, immune suppressants. And the last thing you want to do with a patient with cancer is to immune suppress them even further, because it's going to drive the cancer. So there are 2 case reports in Nature Medicine last year where they gave fecal transplants to patients with checkpoint inhibitor colitis who had been through all the biologics. And again, they're seeing the same sort of pictures as we see, from there to there and from here to here, nice resolution. So there's a big interest in this space, and I think this will be a very interesting area to go forward into. Pouch. Patients who have a long-standing ulcerative colitis end up having pouches, and I'm sure you quite often see these patients in your clinics, in gastroenterology clinics. So after many years of having had terrible colitis, they end up having their colons removed and their small bowel refashioned into a pouch. As soon as you refashion the pouch, the mucosa of the pouch starts to look like colonic mucosa. And as soon as you refashion the pouch, the microbiome in the pouch, which is actually small bowel mucosa, starts to resemble that of the colon. And in fact, there was evidence from Belgium that if you took the microbiome from the patients before they had their pouch colon removed, then you could very nicely predict on the basis of the microbiome who's going to get pouchitis at 12 months after the surgery. So we put a grant into a trial of fecal transplantation for pouchitis as the next thing. So I think that the future here is very promising in terms of manipulation of the microbiome. I hope we won't be doing fecal transplants in 10 years' time. I hope we've kind of got some mechanistic ideas as to what the targets are which we should be developing into some more rational probiotics. It may be in metabolites. Maybe the bile acids are really important in driving all this. And this is a move away from immune suppression, which I think is really important because there are 30 new targets of immune suppression in the pipeline at the moment for patients with IBD. And I think really the focus has got to be now to a more careful approach to human diet studies to get an idea of what it is that's actually causing this dysbiosis and fixing it. Thanks.